Welcome back to the video tutorial. So in this second portion of the video tutorial, I will show you how to submit a HTCondor job for a genome assembly. And the goal of this example is to show you how we can use actual bioinformatics software in an HTCondor job, and also teach you how we can use some specific input files. So in the case of a genome assembly, we will often need the raw FASTQ files. In this example, we're going to be using an E. coli genome. Specifically, we are going to download some data that is available on the NCBI website. So to get to this data, you can look for the E. coli HTCC 25922 strain. So this will bring you to this page on the NCBI website. And if you click on the related information SRA option, you will see some results here. The page might look a little bit different for you if you don't have these filters on, but for this example, we want to be using the DNA. Also, we want to filter by paired end reads and search for the file type FASTQ. So there are 6 data sets that we can download for our example, but in this HTCondor Scenario 2 example, we are going to be downloading just one of them. So let me show you how we would get the link for the download. So we can click on any of these names, and then if you look on the SRA, if you look for the run section and click on the run number, it will bring you to the sequence read archive, and this is usually where we will have direct access to the FASTQ files. And there's multiple ways that you can get to the download link, but I like using the data access tab, and then you can scroll down to the original format. And because we search for paired end reads, there is a forward reads and a reverse read, which is shown by the _1 and _2 fastq.gz file. So you can right click on that, and then you can copy the link address and paste that directly inside of your terminal. So I'm just going to copy and paste the commands directly from the GitHub tutorial to get our data. So first of all, let's make a folder called reads, and then let's cd into that folder, and then let's download the data using the wget command. And just to simplify the names for this tutorial, for the samples in this tutorial, I'm going to rename them with the SRA accession number, but you don't really need to do that if you were working with your real data. Alright, let me clear the screen and type ls so that we can see what we have. So we have forward and reverse reads for this sample over here. You should note that right now our reads are in our home folder directory, but if you were to work on your real data and you have a really large amount of sequence reads, I would suggest you to put those data into a staging folder, because those files are likely a lot larger than what we're using for this example. Alright, so like what we did in the previous video, we are going to take a look at our executable script and our submit file for this scenario. So this scenario goes with the scripts called spades_1_sample. And first of all, let me show you what the executable script looks like. So the executable script is a bash script, so it begins with a bash header. And inside of the scripts is a spades command. So this is the assembler that we're using for this example. And this is the exact command that you would be typing inside of your terminal if you were to submit this, if you were to run this directly from your laptop. But here we're putting the command that we want to run inside of this sh script. You can see here that it takes the forward reads and it takes the reverse reads, and we can say in what is the name of the output folder. So in this case, we're creating a new folder called spades_output. And it also takes as an argument the number of threads. So this is similar to the number of CPUs. And we can also set a memory limit. And then something else I want to mention is when the job is running on one of these remote machines, it will only export back the files in the main working directory. But if there's files in subfolders, then those files are not exported back. So in order to save all of our progress, we know that all the files will be in this spades_output folder. So what we want to do is we want to compress that folder by using the tar command. The -c means compress. And we're going to compress that whole folder into one single tar file that will contain all of our intermediate and final output files. And finally, once we've done that, then we're just going to print to the screen, done. So this is what the executable file looks like. Now let's take a look at the submit file. Let me clear the screen and let's take a look at the submit file. So compared to the first example, there's now new lines that have been added, and these lines, universe container and container image, tells us that we want to use a pre-made container image from Docker that contains the software of our choice. So on CHTC's HTC system, it is recommended to run software through containers, either apt-container or Docker containers. And here we are using a pre-made image, a pre-made container image on the Docker Hub website. So the structure that we will be following is beginning with Docker and then colon 2/ the username of the person or of the organization that made this container, the name of the software that they're using, that they have created, and a specific tag. So what is Staff B? So Staff B stands for the State Public Health Bioinformatics Lab. And they're an organization of public health labs that publishes Docker images for a lot of bioinformatics analyses. So let me show you what the Docker Hub website looks like. Docker Hub. And let's say that we want to run the program spades, and we want to find a container for that. So I'm looking for spades in the search bar, and I press enter. And we can see that there are multiple results for spades, and the part before the / is the username, and you can see that a bunch of users or organizations have created spades container. When you're using a container, it is important to use a trusted organization. So the State Public Health Bioinformatics Community is one of them. So you can click on that name, and you'll notice here that this is the same syntax as here. And I'll show you where we can get the tag number. The tag number is coming from the second tab here. The most recent one is called latest, and then there are numbers here for the tag, so 4.2.0, 4.1.0, and so on. I always recommend using a specific tag number that is not the latest, because the latest version might change depending on when you're running this. So in this case, we want to use Spades 4.2.0. But let's say you want to rerun this code with another version. All that you would need to do is to change the tag number for an available tag number on this website here. Alright, so now that we know what a container image is and how we can use it in the submit file, let's take a look at the other lines of our submit script. So we have the executable, and now it points to the sh script that goes along with the submit file. There's also a new argument that we're using, which is request CPUs. And you'll notice here that request CPUs is a variable that is actually defined lower in this text file, and it has a value of 16. And this is often important to include in bioinformatics job, because it's not because you requested a machine with a certain number of CPUs that the actual software that you're using is going to be using all of them, because some software have a default value for the number of CPUs that they're going to be using. So it is often useful to define that variable and use it in the arguments line so that our sh script can use that value inside of it, the command that is relevant to it. Next, we have the transfer input files. So compared to the first example where there was no real data to work with, now we have FASTQ files that we want to analyze, and specifically we have two FASTQ files. We can transfer input files by pointing to the path or the absolute or relative path to that data. And this is important to do, especially when we are working with data that is currently somewhere in our home folder, because the remote machine that is going to be running our job does not have access to those files. So we have to explicitly write them in to this line. So here we have two files that we want to import. The next lines are the log error and output files. Nothing much have changed here, except that we've wrote a more appropriate log file name. We're using the sample name here. Next, we have one campus pools equal true and one OS pool equal true. And this allows us to connect to machines that are part of the OS pool, sg-htc.org. And you can see that there are many institutions that we can access the computers directly from our submit file by writing want OS pool equal true. And then finally, and this is actually optional, you wouldn't really need this for your script to run. It's just an extra option that's nice to know about. And then there's also the request CPUs, memories, and disk section. Once again, very similar to scenario 1, except that because we're working with a real data set now, we are going to be increasing these values compared to scenario 1. And finally, for the queue statement, we don't have any number next to it because we're only wanting to run one instance of this executable script. So let's submit this file. Okay, our one job has been submitted. And if I type Condor Q, we can see that it's idle. It might take a little bit longer for our job to find a spot to run, but at this point you can exit out of your terminal, you can log out and log back in again to check the results later on. Alright, so it's been a couple more minutes and I'm gonna type Condor_Q to check the status of my job, and it is not running anymore, so it's not in the queue anymore. And once it's done, it will also not show up in this table. So if I type ls, I will see that there's a new file, which is the tar.gz file that gets exported once the job is finished. And you can type ll-lht also, And this is just a different way to like print the contents of the directory, and you can see the timestamp that this was created. So let's go to the logs folder and type lht reverse lhtr, and we can see that there are 3 files associated with this job. And if we look at the first file, which is a log file, we can see that actually a couple of things have happened. So first, we submitted the job at 11:21, and then it automatically starts transferring the files onto this machine over here. But then it started executing the job and it started running, but then the job was evicted. So then it found another computer to run the job, and it started transferring the input files again at 11:23. And then it ran 11:24, 11:24. So this was at the Utah computer. And then image of the job has increased, image of the job has increased, and then it finished running at 11:41 with a normal value of 0. So we can also take a look at the out file, and this is what was printed out to the screen as our job was running. So a lot of spades command here, spades output here. And then we can also look at the error file, and the error file is empty. So let's clear our screen again, and let's take a look at this output folder. You can technically unzip everything here directly by typing tar xf and then the file name, and then you can navigate into folders. But if you want, I'll also show you how to transfer that file locally to your computer. So let's begin by unzipping this here. Alright, so we now have a folder that contains all of these outputs. So probably for genome assembly downstream analyses, the files that you would be needing are the contigs.fasta or scaffolds.fasta file, and these will be your assembled contigs. Let me show you how to transfer your files to your computer. So you can open a new tab, and I will go to my downloads folder. So this is on my computer, not on the CHTC server. I'm gonna make a directory called my output spades. You could name this whatever you want. And I'm gonna use, there's 2 commands that are pretty common to use, the scp command or the sftp. I'll show you how to use sftp. So to use sftp, you type sftp and then your CHTC login. And now the prompt has changed. So you can cd into the folder that contains your the data that you want to move. And if you want to move this whole tar.gz file to your laptop, you use the command get. And this will download it directly to your folder my output spades under your downloads folder. And once you're done with that, you can type exit so that the prompt changes back to this normal prompt here. The other command that you can use to transfer files over is scp, and scp takes 2 arguments. the file that you want to move, and then where you want to move it. So to transfer a file from the server, you type your login, then 2 dots, and then the path of what you want to move. So let's say we want to move just this contigs.fasta file. And then space where you want to move it. And since our current directory is my output space and we want to move it directly here, we're just going to type dot and then press enter. And now if we type ls, we can see that there's the tar.gz file that we transferred using the sftp method, and also the contigs.fasta file that we transferred using the scp method. So that is all for scenario number 2. In the next video, we will look at how we can generalize this script to run it on multiple samples at once.